Okay, so today I am back with you after a long gap. Um, Today we will cover a variety of technologies that are based on uh, DNA hybridization or DNA RNA hybridization. So before I get into the topic, little bit rejigging your memory from your school days, or maybe in the uh, college days if you have studied this subject. Uh, so uh, just a minute. Yeah, so what we need to understand is what is the fundamental principle behind DNA hybridization. So it is possible that some of you may think that it is hydrogen bonding, okay? But uh, it is no longer that recent. At least 20, 25 years back, it was shown theoretically that hydrogen bond cannot possibly account for the uh, strength of the DNA duplex or DNA RNA duplex. The hydrogen bonds do not require that much of energy to break up. What is a more critical force there is what is called as stacking interaction. The stacking interaction is between the nitrogenous bases from the two opposing strands, where the diffused pi electron cloud actually is responsible for the hybridization. Okay. So there, uh, what so what we uh, need to therefore understand that what are the forces that will increase the stability of a DNA or DNA RNA hybrid, and what are the forces that will destabilize these hybrids. So one obvious thing is temperature. Uh, as you go up, the hybrid stability will go down simply because of Brownian motion. The other is that negative charge based repulsion. Do not forget that the DNA or RNA polymers are overwhelmingly negatively charged because of the phosphate backbone. So you can imagine that these two highly negative charges are coming together and staying together uh, is because there is something more overwhelming than the negative charge repulsion between the two strands. But nonetheless, the negative charge repulsion between two strands is a destabilizing factor. So you can actually mask it. The way to mask it, meaning that you reduce the charge-based repulsion. The way you can do it is if you flood the DNA solution with salt, then the counter ion of the salt, that is sodium or potassium ion, that will bind to that will neutralize the phosphate backbone's negative charge, and therefore the stability actually goes up. So DNA strands at a higher salt concentration, DNA hybrid or RNA hybrid are more stable at a higher salt concentration than at a lower salt concentration. This is very different from the protein-protein interaction, which generally falls apart as the salt concentration increases. Okay, and then the factor that favors it, as I told you earlier, is the hydrogen bonding interaction and pi-pi interaction or stacking interaction. Okay, so we will keep this in mind. Now, the first hybridization based technology that became a backbone of molecular biology practice in early 19th century, like mid 19th century, is what is called Southern hybridization. I know that all of you know that it is named after the scientist whose last name was Southern. That's why it is called the Southern hybridization. It is called Southern blot technique. Why is it called Southern blotting? Because what was the objective first? Why did we do Southern blot? Southern blot is to detect the presence of a specific DNA fragment in a solution. Okay. Please try to understand that here you are looking for detection of a specific DNA fragment. You know which fragment you are looking at. Okay. Why is it that you know? Because the whole technology is based on a probe that you make. Okay, so because you know and this probe is actually a DNA sequence or an RNA sequence. Like for Southern blot, it's a DNA sequence. So because you are making the probe, then obviously you know the sequence that you were looking for. So you know the DNA fragment that you were looking for. You just do not know whether it is present or absent. So the way it is done is a sample of DNA or sometimes even DNA protein mixture is treated with restriction enzyme and run on a gel. Okay, it's typically an agarose gel. Okay, after you have run it on a gel, you can look at it under UV lamp to see that the DNA fragments are nicely resolved. Then you transfer it onto a piece of paper. You look at this particular organization here, that there is a filter paper on the very top. It is actually not just one filter paper. I can tell you that we used to put like huge bunches of hand towels, okay, like paper towels. And right below that, you have the nitrocellulose membrane, okay? And below that was the gel. And then there is an alkaline solution. Very important here that we used to take particular precaution to ensure that there is no air bubble between the gel and the nitrocellulose membrane, because if there is an air bubble, that particular spot, no transfer of DNA onto the nitrocellulose membrane will be possible. And then the solution is alkaline. Okay, why is it alkaline? It is alkaline because alkali will denature the double-stranded DNA present in here. Okay, so now when it is transferred onto the nitrocellulose membrane, why is it being transferred? Because of a wicking effect. So there will be a paper here, that paper will get wet, and that wetness will be transferred from here to, and this will act as a sink. Because of this very thick stack of papers, liquid will continue to flow up, and as the liquid will continue to flow up, then along with it, the DNA fragments will also move from the agarose gel to the nitrocellulose membrane. Once it moves to the nitrocellulose membrane, you can actually do an UV cross-linking, or sometimes even baking the nitrocellulose membrane will work. It will cross-link the DNA to the nitrocellulose membrane. So now you have made a replica of your gel on the nitrocellulose membrane, and you have ensured that the DNA molecules that are transferred onto the nitrocellulose membrane, they do not move around or fall off from the nitrocellulose membrane, because you have done a cross-linking. Okay, and the DNA present there are denatured, so they are no longer double-stranded. Why is that important? Because you will be detecting a piece of DNA with a DNA probe. So, and that detection works on the principle of hybridization. So if the DNA molecules are double-stranded to begin with, then its tendency to hybridize with a complementary DNA sequence is satisfied within the complementary strands right here. So they will have no incentive to hybridize with the probe that you are supplying from outside. So this is the reason why it is important to denature the DNA as they are being transferred. Okay? So now, after you have done that, then you, because you know which DNA fragment you are looking at, you make a radioactively labeled DNA. Okay. Now, this is where you need to pay attention. So the radioactivity that is typically used is P32. Okay. Now, P32 is phosphate group. Okay, like it is a radioactive phosphate. But in this particular case, you do not provide the phosphate per se. You provide, sorry, you provide ATP. Okay, you provide ATP, and this ATP will be radio labeled. Now, if you remember, ATP has 3 phosphate groups, of which, so if this is the A part, then it is P, 3 phosphate groups. Closest to the nitrogen base is called the alpha, then beta, and then gamma. In the process of DNA polymerization, these 2 phosphate groups are kicked out. So if you have radioactivity in there, it does not help. So you have radioactively labeled alpha phosphate. So these are called alpha 32P ATP. Okay, so radioactive ATP, but label at the alpha position. Okay, so now when you do that and you make a piece of DNA, you can do that by PCR amplification. Then you can do that by PCR amplification, where along with other nucleotides, you simply use alpha 32P ATP, okay, which will then get incorporated into the DNA that is being amplified. Of course, you have to use all sorts of precaution because radioactivity can spill over and it can be a disaster, etc. Anyway, so once you have that labeled DNA probe, you denature that, because again your PCR product is a double-stranded DNA as well. So you denature that, and then you bathe your nitrocellulose membrane onto that radioactive, with that radioactive solution, and only that DNA which will have complementary sequence to the radioactive DNA will bind the radioactive DNA polymer, and everything else will and no other place there will be hybridization, right? And when you are hybridizing, you want to make sure that you do not have non-specific hybridization. Therefore, initially you will what you will do is you will try to maximize the possibility of hybridization. So you use very high salt concentration and very low temperature. Okay, so this is the maximal permissible hybridization condition. Then you slowly lower the salt concentration and increase the temperature, and radioactive DNA will wash off from any other place of the nitrocellulose membrane other than where it underwent a specific hybridization. In this particular example, it is shown to be here. Okay, and then you take that piece of nitrocellulose membrane, you expose it to X-ray film, and you get to see whether you see any X-ray opaque band. If you do, then you know that the particular DNA fragment that you were looking for was present in this specimen. Okay, so this is what is southern hybridization. You can do the same thing with northern hybridization, like with RNA as well. So if you want to look at a particular kind of RNA that is present in your specimen, you can do the same thing. In this case, you will run RNA sample on the gel. The only thing is that RNA is known to have very strong secondary structure. You see, like DNA is double-stranded, right? But RNA is not double-stranded. RNA is single-stranded. So when RNA is single-stranded, every possible opportunity it gets for intramolecular base pairing. So you remember that intramolecular interactions are much more favored over intermolecular interactions. So because they will have these intramolecular interactions, so RNA will form very complex secondary structure. And if you have such complex secondary structure, the secondary structure formation is driven by hybridization to begin with. Therefore, the RNA sequences are no longer available for hybridizing with an external probe that you are supplying. Therefore, just like here, you have to first denature the RNA. But unfortunately, RNA is completely labile in presence of alkali. Okay, RNA will just hydrolyze. So you cannot use an alkaline solution. So what is done therefore is that a formaldehyde solution is used. So these are run on formaldehyde containing gel, and during the run, the RNA gets denatured. They run true to their size, because like if you have two pieces of RNA identical size, but one is just a linear RNA and another is just a complex secondary structure, this one will run much faster than this one. Okay, just because it will offer resistance to the pores, so it will run a little faster. So to make sure that RNA molecules run true to their size and they are amenable to hybridization, you run this denaturing gel or formaldehyde containing gel, so that all the RNA molecules are denatured when they are transferred onto the paper, and the rest of the process remains the same. After you have done that, for example, if you are trying to look for presence of beta globin mRNA in a specimen, you simply run like say you want to know whether you expect blood cells to have beta globin mRNA, right? But you may not what the system here is that they are using differentiated erythroleukemia cell. Okay, let me split it for you. Erythroleukemia meaning cancer of the red blood cells. So now in every cancer, cells de-differentiate, meaning they go closer to a stem cell like state, so that they may not express the differentiated markers. So differentiated blood cells, erythrocytes, they express beta globin mRNA, but not the undifferentiated ones. So if you take RNA, the total RNA from the undifferentiated cells, and if you treat them, I do not know what is the appropriate condition, but if you treat them to coax the cells to become erythrocytes, you can do that. Many types of cancer cells, you can actually coax them to differentiate, all in vitro, of course. Then they will start to express beta globin mRNA, which is a hallmark of erythrocytes. So in earlier days, just to see if this was working or not, you can take total RNA from the undifferentiated erythroleukemia and erythroleukemia cells that were subjected to erythrocyte-like differentiation for 48 hours or 96 hours, run all the RNA samples on the gel in presence of formamide and formaldehyde, and then you transfer it onto a piece of paper and you do hybridization, because it's just rhyming that the scientists in those days have done. The original one was called Southern Blot in the name of the discoverer, but this one was called Northern Blot just for the sake of it. Okay, so you can actually detect that beta globin mRNA is present. So this is how these two landmark technologies were developed, and they were used extensively to detect a piece of DNA by Southern and a particular species of RNA by Northern hybridization. Nowadays, these are not used very extensively because you can actually run PCR reactions, which will give you specific amplicon for a piece of DNA, or if you do reverse transcription followed by PCR, you will get a specific amplicon for if your particular RNA species is present. You can even do quantitative reverse transcription PCR to give you a fairly good idea about the abundance of the particular RNA species that you are looking at, right? So all of these are technologies that catapulted molecular biology into really molecular terms, but nowadays they are very rarely used. But us developmental biologists, we are critically dependent on one particular technique, which is referred to as in situ hybridization. Now let us understand this term. What does this phrase in situ means? In situ means in its place, okay? And hybridization is, of course, like DNA hybridization, RNA hybridization. So why is it important? Why do we developmental biologists, we use this technology all the time? So we operate by this principle that, see, our objective is still to connect genotype and phenotype, right? So if we speculate, for whatever reason, we will come to all those different lines of investigation later. If for whatever reason we speculate that a particular gene product is responsible for development of a particular species, a particular structure, whether it is, in this particular example, it is about this structure called, these are neuronal structures. Okay? So it is there, you can see it running all along the neural tube, and if you section the head, you can see it is in the neurons. So if I speculate that a gene may be important for neuronal development, okay, then the minimum condition that it must satisfy is that the gene must be expressed in the neurons. Now, when I say the gene must be expressed in the neurons, I mean that the gene product will be formed in the neurons. There are two types of gene products. It can either be an RNA or it can be a protein. Now, to detect protein, you will have to do antibody-based detection. Now, antibody for each and every protein is not there, okay? It is simply not there, okay? And it is much more expensive and time consuming. So a much easier way is to see if the RNA is there. If the mRNA for the protein or the mRNA corresponding to the gene is there. Now, once you have detected the mRNA, if you feel like, if the experiment desires, then you can go for detecting the protein as well. Okay, that is a different issue altogether, but you can do that. Okay. So how do you do it? So now you need to understand that it is not good enough to run a PCR reaction or something like that. Say, if you try to visualize this particular structure, I know it is a little complicated for you at your stage to do that. This is a tiny, tiny structure below what is called the neural tube. Okay, this neural tube, the anterior part or the most frontal part of it is your brain, and the rest of it is your spinal cord. Okay, now in the embryo, it runs right through the middle of your body, midline of your body. There is a structure right below that which is called the notochord here. Okay. Now, you may wonder that why don't we use something like RT-PCR? The problem of that is that when these structures are in the embryos, there is no way you can tell just by looking at the embryo whether it is the neural tube or the notochord or some other structure that you are looking at. And because these are so tiny, you will have to use a surgical technique. And the surgical technique is seldom precise. How can you know that you surgically collected only the notochord and not cells from here or here? Because once you have collected an isolated total RNA, you have isolated total RNA corresponding to the tissue you have excised. You have no way of controlling that you have excised only this part of the tissue and you are looking at that. This is the reason why you use this term called RNA in situ hybridization. Like, we will look at the RNA in its own place, meaning where it is expressed. And this is where we basically use the embryo just like the membrane in the southern or northern blot. What do? We harvest the embryo and then immediately we put it in a solution of paraformaldehyde. Okay? Paraformaldehyde, what it does is it fixes the embryo, that is, introduces small crosslinks between RNA and protein and protein and everything. So it actually inactivates all the proteins present in the embryo. Those proteins include proteases and RNases. So that way, the tissue will not rot and the RNA will not get degraded. So that's why it is called fixing. So exactly the state in which the RNA was present at the time of harvesting the embryo, that will remain there permanently. After that, we do all sorts of processing, but eventually we will, RNA is single-stranded to begin with, right? So we will probe it, but this time not as a DNA probe, which is done in northern hybridization. We have to actually probe with an RNA probe, but we have to go for an antisense RNA probe. So if you know the sequence of the mRNA with the poly tail, you will design a probe somewhere from here, and it will be 5' to 3'. Okay, so this is called an antisense RNA probe. This is called an antisense RNA probe. Okay, now this antisense RNA probe, again, just like we used alpha 32p ATP for southern or northern hybridization, here we use nucleotide that is decorated with a chemical called deoxygenin. Okay, so this is a chemical that is appended to the cytidine triphosphate, okay, CTP. Okay, and this is then used in a reaction for this is used in a reaction of in vitro transcription. So what is in vitro transcription? That is, you make the RNA in the test tube, this antisense RNA. How do you do that? Go back to your, let me go back to the cloning plasmid slide, okay, just for a minute. So, remember that this cloning plasmids, they will have these T3, T7, sp6 or M13 primer binding sites, of which T3, T7, sp6, they are basically names of RNA polymerases from bacteriophage. And these RNA polymerases, they have very short, 20 to 22 nucleotide long recognition sequence. Okay, and these sequences will be there on either side of the multiple cloning site. Okay, so somewhere over here you will have the T7 sequence, and somewhere over here you will have the T3 or sp6 sequence. Now in the multiple cloning site, you have cloned your favorite piece of DNA for which you are going to do the RNA in situ hybridization, right? Because you have cloned it and you can sequence it, or you know the restriction site orientations, etc. So you know which is the sense strand and which is the antisense strand. So if you want to make the antisense of the sense strand, then what you will do is you will use T3 or sp6 RNA polymerase. If you do that, then it will bind to the RNA polymerase will bind to this recognition sequence present right over here, and will start to crawl along this strand. So remember, this is 5' to 3'. It will take instruction from this strand and will start to make the new RNA in the 5' to 3' orientation, and that is your antisense strand. And as this transcription is happening in vitro in a test tube or in a small plastic tube, you will have to supply your own RNA trinucleotides, okay, like ATP, GTP, CTP, and UTP. So when you do that, then you will make your antisense RNA. Sorry, let me go back to that slide. So you will make your antisense RNA, which will have incorporated CTP with deoxygenin. Okay. Now there is an antibody against deoxygenin. Okay, it's called anti-dig antibody. Okay, so that antibody, if you now put it in the so the antisense RNA will hybridize to the sense RNA that is present in the embryo wherever it was expressed. So the antisense RNA will then hybridize onto it. Now you want to know where the antisense RNA hybridizes because you want to know where the mRNA is expressed. So you will then probe it with the anti-dig antibody. But the anti-dig antibody by itself is also not visible. So then you use this anti-dig antibody is typically produced, like say in goat. Okay, I'm just making it up. So then you will use an anti-goat secondary antibody. A secondary antibody is something that is used to detect the primary antibody. In this case, this is the primary antibody. The anti-dig antibody is the primary antibody. These secondary antibodies will typically have an enzyme coupled to it called alkaline phosphatase, or it can be something else. Now, this alkaline phosphatase enzyme, if you provide it with a pair of substrates, which is NBT and BCIP, okay? So BCIP is basically a phosphate, which is a substrate of alkaline phosphatase. And in the presence of NBT, when the phosphate is removed from BCIP, then a blue colored precipitate is formed, and that's what you get to see in the RNA in situ hybridization. Okay, so when you see this kind of pattern, you know that the antisense probe that you used is against an mRNA that is expressed in the neurons like this, and the antisense RNA hybridized to it, to which the dig antibody bound, to which the secondary antibody with AP coupled to it bound, and that's where the precipitate formed of BCIP after dephosphorylation. So this is an indirect method of detection, but it is a very, very powerful method, and every developmental biologist around the world still use this technology. I would not say every, like this technology is not developed for C. elegans, for example, but mouse, chick, zebrafish, plant, frog, in all these kind of species, RNA in situ hybridization is a very popularly used methodology, and there are two variants of it. One is called whole mount, and another is called section. So whole mount RNA in situ hybridization or section RNA in situ hybridization. The applications are different. In whole mount RNA in situ hybridization, you want to know the general organ level presence, whether the RNA is expressed in the limb, or the RNA is expressed in the head, or RNA is expressed in the heart. That you can detect by whole mount RNA in situ hybridization. This is the first level of investigation, less laborsome. You simply harvest the embryo, fix it in PFA, paraformaldehyde, then like you dehydrate it in presence of ethanol, and then you rehydrate it, and then you do the hybridization. Okay, but it does not give you cellular level resolution. Like you cannot get this level resolution from this. Here you only get to see, a faint staining here. So you know that it is somewhere there in the neuronal system, but you have no clue where it is. Then you, like once you know that it is there in the neural system, then you take the embryo and section it. And you do the in situ hybridization after sectioning it. Okay, and then you can get this kind of images, and it gives you exactly in which cell type it is expressed. And I will show you later, there are much more sophisticated methods now where you can do RNA hybridization for 2-3 genes simultaneously. So you can know, like if there are 3 subsets of cells, A, B, and C, you will know that one RNA is present here, another is present here, another is present here. You might find one RNA that is present in both, another RNA that is present in both. So you can get that kind of cellular resolution by doing RNA in situ hybridization. Okay, so these are the 3 hybridization based technologies that I covered today: Southern hybridization, Northern hybridization, and RNA in situ hybridization. They both, all these 3 depend on the same thing: hybridization between 2 polymers of nucleic acids. It is just how you label them varies. In Northern and Southern, you label them with radioactivity. In RNA in situ hybridization, you label them with deoxygenin modified nucleotide. Okay, so that's where we end this class, and we will get back together again for another class in a short while. Okay, thank you.